So let me welcome up to the stage Manpreet Singh, an assistant professor of psychiatry. I'm planning to open my email one of these days, very soon, and read about your promotion to rock star, and the director of the Pediatric Mood Disorders Research Program in the department, and my wonderful colleague Ian Gotlib, who's a professor of psychology, and believe it or not, he is transitioned from being the chairman of the Department of Psychology just a few days ago, and is chosen to spend time with us before going to sabbatical, so you get extra points for that. Come on up.
Thank you very much.
Yeah.
Thank you, Laura. What I want to do today is to, with Dr.Singh, who will come up shortly, we have like 18 minutes. I'm going to take 9, but I've been here since 7:30, as you all have, and as stimulating as it's been, it's also been long. So I'm actually going to try to talk for like 5 minutes and then talk with you and take questions and try to show you enough of what we do to get a sense that you can have questions, that we can then just kind of talk with each other. So I'm not going to do this. We've now had like 5 or 6 presentations of how depression is horrible and growing, and what I want to do is, we talked about suicide. I want to focus on why we started looking at adolescence in trying to understand depression. And so adolescence is probably the peak period of risk for depression. Depression and suicidal ideation or suicidal thoughts are really important precursors to suicidal attempts, most of which occur, those first attempts, in adolescence. As you can see on this graph, this is the cumulative prevalence of depression, and you can see through adolescence it's up to 25%. You superimpose suicidal ideation and suicide attempts on this, and you can see that this period of risk is severe and getting worse. And we're not just shouting in the dark or in this room. This has made media for the last couple of years now. New York Times, Washington Post, all sorts of other fake news outlets have kind of talked about this notion that teens should be screened for depression. They've talked about suicide rates climbing. But actually here in Stanford at Palo Alto, we've had the Centers for Disease Control come because we had a suicide cluster here that they were investigating a couple of years ago. Just last year, we had another high school student here. So it's hitting close to home. And what we were doing in my lab, and with Manpreet, and I know she'll talk about this as well, is trying to identify those children who are going to develop depression. Because if we can identify them, get that first episode detected before it occurs, then we can prevent a lifelong cascade of adverse consequences. So the strongest predictors of developing a depressive episode are having a family history of depression and being female. As many of you probably know, females have twice the prevalence of depression as males. What we did then is to recruit 200 mothers and daughters. These are mothers, 100 of them had a history of recurrent episodes of depression. The daughters were all clean. That is, 10 to 14-year-old girls came into the lab. We diagnosed them. They'd never had yet a depressive episode. And so what we wanted to do is to measure everything that we could measure. We did imaging and cortisol and immune function, and I'll show you some of those data. And we thought that we would then have to wait for 4 or 5 years till some of them became depressed, and then we could see what factors are predicting that. But I will tell you that pretty much everything that we measured in these never depressed daughters of depressed mothers, never depressed daughters of non-depressed mothers, distinguish these two groups of girls. So these daughters who had not yet been depressed but had a depressed mother, higher levels of stress cortisol, higher levels of diurnal cortisol, greater amygdala activation to stressful stimuli in the scanner, smaller hippocampal volume, 12-year-old girls, smaller hippocampal volume. Not surprising given the higher levels of cortisol. that are probably circulating that serve to prevent synaptogenesis in the hippocampus primarily. We said we were going to look at telomere length. So telomeres, as you may know, are they're the ends of DNA. They're like these caps on your shoelaces, and they get shorter as we age, and they're involved in cardiac disease and progression of age-related diseases, but they're also stress-related. And when we said we're going to look at these telomere length in 12-year-old girls, people just said, don't waste your money. With Owen Wolkowitz at UCSF, we did it anyway, and it turns out that 12-year-old daughters of depressed mothers already have shorter telomere length than 12-year-old daughters of mothers who have never been depressed. Mitochondrial DNA, all sorts of other measures. And now, 10 years later, 60% of these daughters of depressed mothers have developed an episode of depression. And these same factors at the tail of the distributions are predicting the onset of disorder. So, one more data slide here, and that is we were also interested, as I mentioned before, in suicidal ideation, and it turns out that in this sample and a similar sample, we can measure brain activation, connectivity before suicidal ideation develops, and we can predict which kids are going to start to develop suicidal ideation, one of the major precursors to depression. The bottom line is we started doing all of this, and I have to thank, I don't think Tom Insel is here anymore, but in his couple of lives ago, he was instrumental in funding this research at NIMH. But it seemed to us that we're too late, that 9 to 14-year-old children, these daughters of depressed mothers already have all of these characteristics. They haven't yet developed an episode of depression, but they already have this elevated stress reactivity. We are just late. And so we started last year to study babies, and not just babies, but pregnant women before they had their babies. This is Kate Humphrey. She was a postdoc in my lab, now an assistant professor at Vanderbilt. Her daughter, Alice, who has now had more scans than probably anybody else in this room. And we started 2 projects that we were able to fund through NIMH and NICHD, assessing maternal characteristics like maternal depression and history of maternal maltreatment, but also infant neural characteristics, endocrine characteristics, cortisol. We're measuring cortisol in hair of the mothers now because hair gives you 5 months of cortisol, month-by-month averages. And so if we measure cortisol in the hair halfway through pregnancy and at the end of pregnancy, we get month by month for 9 months the intrauterine environment for cortisol. And I want to show you, these are the scan data from newborns. They're just very cool. I'm sorry, this little things get me excited. We have structural scans, segmented structural scans, functional connectivity. We can measure resting state in these infants and ASL arterial spin labeling to get cerebral blood flow. I just want to show you some data just to show that we didn't do it for nothing. Prenatal stress, smaller infant hippocampal volume, right? Greater exposure to intrauterine cortisol, smaller hippocampal volume of the infant. Offset by mothers who are more sensitive in their caregiving interactions, and it turns out that that's good. We get stronger functional connectivity with amygdala in infants whose mothers behaviorally in infant interactions are more sensitive. We put a little recording device in the infants, and we send them home, and for 16 hours, it just records everything the infant hears. The more words the infant hears, the better development in their language areas. Maybe not surprisingly, but these are like week-old infants, and we're already seeing these kinds of effects. Here's the last thing I want to show you, and these data, I'm guessing, may not hold up. Don't take pictures, because I don't think they're reliable, but they're enticing. And it just showing you that infant hippocampal volume related to family income. Now, the infant doesn't know how much money the family has, for sure, so it's a proxy, but for what? I don't know. So if I can, I'm going to just turn this over. I failed. I'm sorry I got too excited. Let me introduce Dr. Singh.
Maladaptive behaviors and
I
thoughts,
thought I could do it.
when they become embedded into our daily lives, become very challenging to treat and reverse. I hope we're beginning to convince many of you that we need to start identifying these problems early and treating them early. So I'm going to take the baton from Dr. Gotlib and tell you a little bit about how we can be more successful in developing treatments that last and that are durable and are lasting to the point of potentially preventing depression, bipolar disorder, and other mood disorders from lasting a lifetime. Ponder for a moment where you get your best ideas. I get my best ideas from my clinic. My patients and families come to me with problems very typical. Some of them are very simple problems, others are more complex. They come to me in distress, often because of familial or environmental challenges, parental divorce, transitions to college. And as a clinician, I have to ask myself several questions. What do I do to help my patient? What's the first thing I should be doing? Should I be evaluating for a other medical problem that might be contributing to the behavioral problem? Should I assess for family history? Because as Dr. Gotlib said, family history matters. It's an extremely important risk factor and predictor of intergenerational development of mood problems. Should I assess for safety and hospitalize this patient? Or do I call his therapist, his psychiatrist, and find out what's worked, what hasn't worked? I also ask myself the who, what, where, when, why. Who benefits from the treatments that I will provide for them? Where and in what setting might that treatment land well? The brain is a central player in this system that we're evaluating and treating. And it's complex. It's the most genetically determined organ in the body and is extremely sensitive to the environment. We know that even the fundamental issue of sex, whether you're male or female, has an impact on your brain development, and that the brain isn't just male or female. In fact, a recent study that Dr. Gotlib and I conducted actually demonstrated that the brain is not just male or female, that regions of the brain have degrees of maleness and femaleness, and there are genetic implications for that too. So if we have this complex organ, how are we to imagine that one particular treatment is going to do wonders for many? One size doesn't fit all, and my colleagues in precision medicine are eagerly engaged in this question. The other question that we ask is when? What's the critical sensitive window? In childhood, there are many factors that contribute to the development of a healthy brain. Lots of external factors, innate factors, and combined with some sort of stress or challenge, you either develop intact mood cognitive function or you have some dysregulation. And then you present yourself to a psychiatrist like myself, and either you transition with intact functioning, perhaps you have some resilience, or you might be more vulnerable. And then this is the embedding that can last lifelong. And transitions to college are definitive periods. There are times when kids are really either beginning to experience the full phenomenon of a mood disorder because it's the most common age of onset, or they're struggling to become adults and individuate. And yet, how many interventions do we have targeted for that particular population, inspired by those unique challenges that they are facing? The other day I was listening to NPR, and they were talking about textbooks and how expensive they still are for college students. How come we haven't solved that problem yet? Why are we exposing our children, our transitional age youth, who are now, we're recognizing the rental car companies got it right, that brain development doesn't stop miraculously at 18, right? That's why we don't rent cars to 18-year-olds. But we have an opportunity. In adolescence, we have an opportunity to potentially make a difference. And perhaps by adolescence, as Dr. Gotlib says, it's too late. Maybe we need to even go younger, as early as preconception, perhaps. But this window and understanding timing is going to be critical for the next generations of treatments that we develop. My colleagues and I are also very preoccupied with the why and the how. Some of us are very preoccupied with, well, how does this work? What's the mechanism underlying a mood disorder? Others are, don't bother worrying about that. We know a treatment works, let's just implement it. I think science needs to incorporate both of those concepts to some degree. We need both some ambition, and we need to try treatments in people and see if they work or don't work. And we have to understand why they work when they do work, or why they don't work when they don't work. Ultimately, Tom Insel actually wrote a great article on translational psychiatry a few years ago. It's almost been a decade since he's wrote this article. And I think that the 3 principles that he highlighted in here are critical and very relevant to today's conversations. We need 3 major breakthroughs in psychiatry. We need biomarkers for early detection, preemptive interventions for those detected to be at risk, or those in pre-symptomatic stages of mental illness, and we need better treatments for people living with symptoms that predict and track outcomes. That's probably why a bunch of us who are very interested in this space, in this developmental space, got funded from the NIH, because there was some clear importance to this issue. If you look at trials, clinical trials that have been done in thousands of children and adolescents to date, there is a massive placebo response. And curiously, it's very, very high in depression. In pediatric depression, you see very nominal separation between active treatment and placebo response. Curious. Why is that? What is it that we're doing when we bring children in for clinical trials and give them a compassionate ear to listen to? How can we leverage that? We have an opportunity to potentially understand the neuroscience of the placebo response. When we do clinical trials and we incorporate neuroimaging pre-post intervention, we should look at what happens to the brain in response to a placebo and try to design studies that are based on that information. Another challenge we have in the treatment of children and adolescents with mood disorders is that we have a hard time distinguishing symptoms from side effects. Have you guys ever seen the commercials from pharma that show you a particular psychotropic medication and it's indicated for sleep, depression, bipolar disorder, whatever the case may be, and then you look at the fine print side effects? What are the side effects? Sleep, depression, mania. How do you distinguish symptoms from side effects? We have yet to do a definitive study that helps us understand this fundamental concept, and yet we've done thousands of clinical trials to date. We have learned in our pilot work in the past 20 years that, in fact, there are certain populations of youth that are much, much more vulnerable to side effects compared to the general population. These are children with a family history of bipolar disorder. They have about a 50% or 10-fold risk of having an antidepressant-related side effect compared to the general population. Symptoms can appear immediately, within weeks, or even randomly a year later. And we have a hard time distinguishing the symptom from the side effect. Another important and compelling point to make in terms of this pediatric and developmental space is that if you look at this nomogram, it shows you that if you are under the age of 12, your chances of having a side effect, especially if you have a bipolar family history, approach 100%. For this reason, we were funded to do a clinical trial to evaluate the safety and tolerability of Lexapro, of escitalopram. We know that it works in kids. We've done the randomized controlled trials. But do we know that it's safe? We have, in partnership with the National Institute of Health and the Data Safety Monitoring Board for NIH, looking at this question very critically. Because not only is it affecting a population of youth that might get antidepressants and not have a good outcome, but there are biological implications. Those same youth who have developed a side effect compared to those who don't have smaller amygdala volumes and amygdala hyperactivity. This next slide is describing the nurture effect. Title of my new favorite book. Everyone should read it, by Tony Biglin. He's a prevention scientist. And the point I want to make in this slide is that we actually can do a lot to make society better and to improve the lives of youth who are struggling with mood disorders and other psychiatric conditions. We've discovered through collaborations with Dr. Gotlib and designing studies looking at children of parents with mood disorders, including bipolar disorder, that in fact those same youth are not just at risk for all of the biological complications that Dr. Gotlib described, but that those connectivities in the brain are impacted by their environmental chaos, their family chaos. The more the chaotic the environment, the more disconnected the brain. And in fact, we've also found and discovered that if you treat those same youth with family-focused therapies, interventions that are inspired to improve education about their symptoms, inspired to help them regulate their emotions better and problem solve effectively as a family, their prefrontal cortical function improves. And that improvement corresponds to improvement in symptoms. Finally, the last couple of slides I want to share with you just describe to you how we're striving for mind-body harmony. Kids are not just susceptible to brain-based problems, but those brain-based problems have an impact on their peripheral body. The brain is, after all, the conductor. And so we're evaluating youth with depression and obesity. Obesity is becoming an epidemic in children. How can we address that issue? And is there an opportunity for us to potentially prevent the development not just of suicide, but of cardiovascular disease and diabetes by addressing youth early in development? Because guess what? When you look at youth who have depression and obesity, they already have developed insulin resistance. And their brains are showing evidence of dysregulated reward function corresponding to peripheral insulin resistance. Finally, because we believe the brain is a central player in this system, we are beginning to target that system for youth who are particularly challenged and who have failed multiple antidepressant trials. We're giving them transcranial magnetic stimulation as a treatment alternative. This is part of the first multi-site randomized controlled trial to evaluate the safety and efficacy of TMS in youth. And we're hopeful that this will lead to an FDA indication for a certain portion of youth who are particularly vulnerable and not responding to traditional treatments. With that, I'd like to end and appreciate your time.